And after this, we obtain a whitish pellet with a transparent supernatant. The supernatant that contains the plasmid or DNA is transferred into these columns that have DNA binding affinity. Now in this step, it's important to transfer the supernatant without disturbing the pellet. After this, again these columns are centrifuged for a minute and the supernatant is discarded. Next, this buffer PE, which is wash buffer and contains ethanol, will be added to the column, and the columns will again be centrifuged for a minute and the flow through will be discarded. To remove any kind of residual buffer left, a second round of spin is given, and next the column is transferred into a fresh append of tubes. Next, these columns are incubated at 37 degrees Celsius so that the residual alcohol gets evaporated. For elution, this buffer EB or autoclave water heated at 60 degrees can be used. It is important to add the buffer or the autoclave water in the middle of the column and not on the walls. And after a minute or two of incubation, these columns are centrifuged. Now this time the flow through will contain the DNA, and the concentration as well as the purity of the DNA can be checked by nanodrop.